Elissa is a fifth-year PhD candidate in the cortical neural prosthetics lab at the University of Michigan, and she will talk about mechanical modifications to carbon fiber electrodes increase recording yield in brain and enable interfacing with peripheral structures.
Yes, hi. Can everyone hear me? Yes, okay, awesome. So hello, my name is Elissa Welle. I'm a PhD candidate from the University of Michigan in Professor Cindy Chestik's lab. And I'm here today to talk to you about our carbon fiber recording electrodes and the modification of these electrodes for improved brain and nerve interfaces. So this work is based in the field of neuroprosthetics, which translates signals collected from the brain, from nerves and muscles into mechanical movements of external robotic devices or into the stimulation of existing musculature that may no longer be receiving neural input. And brain-machine interfaces is a well-known technology that incorporates neuroprosthetics. The signals from the brain are recorded, decoded, and then translated into the movement of a computer cursor or robotic hand. Patients like Kathy, seen here, who is left quadriplegic from a brainstem stroke, are able to regain some independence, like being able to feed herself a cup of coffee. So this technology has a potential for a very far-reaching impact, as there are currently 6 million people living with paralysis in the US today. And the crux of this technology, and also its limiting factor, is the electrodes used to record from the brain. So the only FDA-approved cortical electrode is the Utah array, seen here. It's a 96-channel device. It's been around for decades and successfully lasts in patients for years. However, it's fairly well known that conventional silicon electrodes in the brain cause scarring. And our lab was given a rare opportunity to do histology on a single non-human primate implanted with a Utah array for roughly 2 years. And while this data is very preliminary, we can obviously see scarring caused by the Utah array. This is just one slice along the shank, not necessarily at the tip of the shank where the electrodes are. What we're seeing here is endemic throughout. And if you look at the electrode hole itself, one electrode hole, there's an obvious difference between the number of neurons, which are shown here as yellow dots, around the electrode hole as compared to the control tissue. So the data is still being processed. This is very preliminary, but our main takeaway here is that there are just many fewer neurons that we might have otherwise had access to record from. And so the question is, how do we go about maintaining a healthy neuronal population around the electrodes? People have come up with many alternative styles and types of electrodes to solve the scarring problem that we see with the Utah array. Our solution is small electrodes. In our lab, we work with carbon fiber electrodes specifically, each with an individual diameter of 8 microns, once coated with parylene insulation, with a 1 kilohertz impedance of less than 100 kiloohms when coated with PEDOT PTS. And so carbon's a really unique material. It's electrically conductive, it's chemically stable, it has high tensile strength, and also very good fatigue resistance. And I'll talk some more about that later. But one of its best properties is its subcellularly sized diameter. And so, you think that maybe these are too small to insert. They're only 8 microns. But because of its inherent stiffness, carbon fiber is able to insert with very minimal assistance from a dissolvable PEG coating. And so we can insert these tiny fibers into the brain as an array structure. And when we reconstruct a 3D image of the tissue around the fibers after they've been implanted for months, this is in rats, we reconstructed it by slice. And this video is showing 2 carbon fiber electrodes traced, and you can see them here in gray from 2 angles, and the neurons are in green. And there's a really dense neuronal population right near the electrodes. That you can see neurons, cozied right up near the electrode tips, which means that these electrodes are getting many more neurons to record from for future brain-machine interfaces if we were to use these fibers. So despite this very encouraging histology, there's still work to do. And we noticed that when we implanted these chronically, when we implanted functional electrodes, even though their impedance remained really good, we are actually only recording from roughly 30 to 50% of our electrodes, our functional electrodes, in the brain. So we implanted them and only 30 to 50% the functional electrodes recorded any neural units. And so, this was confusing because despite the good histology and the low impedance, we're only getting about a third of our electrodes that are actually recording spikes, which is obviously the point of what our electrodes are for. So, in order to use these in future brain-machine interfaces, we want to increase this recording yield to hopefully 100%. And we took a look at our fabrication methods. So there's more about this in our papers, but briefly here, you take a bare carbon fiber, we coat it with perylene C, as I mentioned before. We then cut the tip using microsurgical scissors, and then we electrochemically deposit PEDOT PTS on the exposed carbon. And when we took a look at these traditional scissor cut electrodes under SEM, however, the results were not great. We saw that the carbon was really inconsistently exposed from perylene. The sites were rough. It was overall highly variable. And, we felt like this was a first and necessary step to improve upon. So we developed a method using blasts from a green light laser to expose the tip of the carbon fiber and peel back some of the perylene to create a more consistent electrode site. You can see some SEMs here of the device or of the electrode with a more consistent and variable and less variable exposed carbon. And so the laser exposes roughly 10 microns of carbon from the tip to create a surface area of like 250 microns squared. And then the impedance drops down to 20 kilo ohms when coated with PEDOT. And so, we've created an electrode that does have a greater surface area than previously seen before. But these dimensions are still on par with an ideal point source electrode. And in fact, the size of a neuron in layer 5 of a rat motor cortex is roughly 15 microns. So we're still, well below the size of a neuron. And, we've created a subneuronally sized electrode with very low impedance. So to test and see if these laser cut electrodes record from neurons and hopefully recorded from more neurons, we chronically implanted 45 electrodes in 4 rats over several months. After 7 days, 84% of the electrodes recorded units. And after 2 months, we were 71% still recorded units, both of which are much higher than the 30% we were seeing from the scissor cut electrodes. So we saw, nearly a doubling in recording yield. But this is not just about quantity, also about quality. And we were, happy to see that the units recorded by the laser cut electrodes were recording amplitudes up to 600 microvolts peak to peak, which, is quite high. And we got, saw that nearly 10% of our electrodes are recording an amplitude that high. And so, with these promising brain results, the natural next step is to translate them into the peripheral nervous system. So right now, again, similar to the brain, the conventional electrodes used in the nerves is the Utah modification of them, the Utah slanted array. And similar to the brain, there's a lot of scarring and inflammation that results from this electrode when chronically implanted in nerves or peripheral structures, and perhaps even more to a severe degree, given this fragile nature of peripheral structures. So, and then also the recording electrodes are so big that recording from like individual axons, where axons are in the single digits of microns in size, essentially it's out of reach to record from individual axons. So, the natural next step is to apply our carbon fiber technology to the peripheral nervous system. So in that process, the first challenge was to be able to actually insert these 8 micron fibers into a nerve. We took inspiration from methods used by the Gardner lab and sharpened the fibers. So the trick here is all about visualization. And I'm going to play this video again so you can see it twice. But we submerged the array underwater in a water bath. The water bath acts as a flame retardant, and we're sharpening them with a small butane flame. And what you're seeing here is actually we're looking at the reflection of the array underneath the water surface so that we can align the tips directly to the water surface so that they can be sharpened by a small butane flame while the rest of the array stays intact. And the ripples of the water that you saw there briefly were the movement of the butane flame. And then afterwards, this is the alignment step. And so that's where the water is moving in response to the flame. And afterwards, you can see a slight tapering in the carbon fiber tips. And so through this process, we get sharpened fibers. There's a really smooth transition between the carbon and the insulation. And although the surface area is much larger than before. Before we were looking at like 10 microns of exposed carbon in length. Now we're looking at 140. You'll see that we still are able to record neural activity in peripheral structures. So we sharpened 200 micron length fibers, very short fibers, on our existing brain array for acute insertion and recording from the rat cervical vagus nerve to test this initial sharpened technology. And what we're looking at here is an array of 16 sharpened carbon fibers inserting easily into a rat cervical vagus nerve. The nerve is elevated from its cavity. And then for reference, a rat cervical vagus nerve is anywhere between 200 to 500 microns in diameter. So given that these lengths fibers are 200 microns in length, we can assume they're landing roughly in the middle of the nerve. And as I mentioned, the surface area is much larger than what we used in the brain. However, the sharpened carbon fibers recorded multiple neural clusters from the vagus nerve. And we also tried these arrays in feline dorsal root ganglion and found that they inserted easily despite the thicker covering of the sheath, and recorded neural clusters in response to brushing. So with all that, one of the next things was that the main limitation of the arrays that I showed you before is the fact that the fibers break really easily at the end of the printed circuit board. So to counter that, we embedded the device partially the fibers in silicone. And the silicone greatly increased the robustness of the carbon fibers. Here you can see glass capillary running over the free end of the fibers without breaking. We went up to 50,000 cycles. There was no breakage at that. We stopped then because there's no breakage. But this shows the good fatigue resistance qualities of the carbon fibers I mentioned before. And this is something that is very useful when talking about chronically implanting these devices in peripheral structures where there's lots of movement within the body. Combining these things, we've created a new array appropriate for chronic recording from peripheral structures. It has fibers partially embedded in silicone, and the top of the interface is only 1 millimeter by 2 millimeter in size. So, it doesn't look pretty, but it works. And it's mixing, and these sizes make it really suitable for recording even from very small nerves in rats. And so finally, we tested the recording ability of this new array that combines silicone and then the sharpened fibers. With our acute device, we were able to implant to a depth of 1.2 millimeters in the rat motor cortex without any assistance from the dissolvable PEG. We recorded neural units. And then we also recently just recorded our first acute spontaneous spikes from the DRG using this new technology. And then our next step is to work towards the chronic implant of these devices in both animal models and assess recording and histological viability. So with that, thank you to funding and then to the many members of my lab and Professor Cindy and Tim Bruns' lab. And I will take any questions. Thank you.
Thank you very much, Elissa, for this great talk. I really love the pictures from your 2020 paper. It's really cool. While we're waiting for questions, I should also mention that YouTube viewers could also post questions in the chat window to the right, and we will paste them here. So, okay, we have the first question. What was the longest implantation time in the rat brain while the electrodes were still recording?
Yeah, that's a great question. Yeah, so the longest implantation time using, so this is without the sharpening technique, this is using our traditional one, is up to several months, so up to 3 months. And then previously up to, 3 months is the longest that we've done. And at the end of that, we were still recording units and we sacrificed due to histological metrics and timelines. Yeah.
I had a question, a couple of questions actually.
Okay.
So how much of the area does it record from? So how local is it, or how broad is the signal that it captures?
Yeah, that's a great point. So I mean, typically we, the recording distance or the recording area decreases kind of 1/r, so 1/r distance away from the recording. So we typically say that, we want to record from neurons within 50 microns of where the recording site is. And so obviously having a much larger surface area means that we are averaging across a much larger area, right? So if we have a huge, 140 micron surface area, like you saw in the sharpened fibers, we're recording from, a huge volume around that around the exposed electrode. So, the ideal point source electrode would be a single dot, and we're able to record exactly from that space. So we definitely are working towards being able to modulate how much exposed surface area there is for the sharpened carbon fiber so that we can get a smaller area. However, we also were encouraged to see that we were able to record neural clusters, maybe not neural units, but neural clusters from the nerve, which means that we are getting accurate and high content information, even though we are most likely recording from a much larger volume.
Thanks. Also, like, can you have multiple contacts on the same electrode? Like you have those silicon U probes, right? Is that possible?
Yeah. So yeah, that's a good question. So each carbon fiber acts as a single wire. So it really allows for just like one contact at the end of it. But there are ways and people in the past have worked on ways to like bundle them together to create a structure that has multiple fibers, which can have, different lengths and things like that. Yeah.
Thanks.
Thank you.
Okay, we have two more questions. The first is again from Ksenia Kirilova, who asked the previous question.
Okay.
Is this flexibility of the electrodes that you showed going to be enough to account for brain movement inside the skull during chronic implantation? And how many channels maximum were you able to implant in one animal?
Great questions. Yeah. So the number of channels is up to 16 right now. So that's in the brain. We've done up to 16 and at one time. And then in terms of the flexibility, yeah, I think that when we implant them in the brain, we do cement them to the skull. So the micro movement or like the micro motion that we'd see is mostly, the most of that is absorbed in the actual array itself and not the fibers. The fibers are implanted into the brain. And so then the actual like flexible polyimide device that the fibers are attached to are kind of what captures the movement of that like brain micromotion. However, the flexibility that I showed with the silicone embedding, that's going to be more useful for the peripheral nerve interface where we have the only thing inside the nerves is the carbon fiber itself. And so as there's maybe some movement between muscles or in, kind of the twisting or shifting of the nerve itself, the fibers will be able to bend and accommodate that without breaking. And so that was kind of the main discovery of having the silicone is that we can have the actual electrodes themselves are able to bend and move and still be robust and not break in response to that movement.
Okay, we have one more question from Don Taylor. Are you
Okay.
working with manufacturers to make this available for purchase?
Yeah, so there's a distribution grant that we have for the brain electrodes, and that's through the Neuromatch Mint Hub. And so if you want to contact, you can contact Paras Patel if you're interested in learning more, receiving those devices for the brain electrode specifically. Yeah,